It is my pleasure to welcome you to the last video in this 10-part online training program on adaptive designs and clinical trial simulation. We have made it to part 10. We are ready to wrap things up, and in this module, in this video, we will apply the same general approach as before. This module accompanies part 9, where we introduce the relevant statistical details and the decision-making framework for adaptive population selection trials. This module introduces the software tool designed for this particular class of adaptive trials. As before, we will begin with the general information on the software tools we have built for a simulation-based assessment of popular adaptive designs used in Phase 2 and Phase 3 trials. These tools, available within the Mediana Designer package, are designed to help clinical trial sponsors and research teams design adaptive trials with data-driven decision rules such as futility stopping and population selection in the context of this particular module. This slide lists the relevant links. That includes a link to the CRAN website from which you can download and install the Mediana Designer package, information about where the technical manuals could be found, as well as a general description of the relevant statistical methods. And the last point, the last bullet point on the slide, is a reference as a link to the online manual with multiple useful case studies that will help you come up to speed with this R package. The software tools I mentioned on the previous slide could be run on your computer, or if you prefer, you could also run the software for a clinical trial design in the cloud. You could do this using the web applications we have released, and this slide shows how you can access these web applications, and that includes the link to the cloud platform where those web applications are available, as well as the login and the password. Now we will focus on adaptive designs that enable population selection at an interim assessment. This slide provides information about the function included in the Mediana Designer package. This function is called ADPOP-SEL, which obviously stands for Population Selection in Adaptive Designs. I would like to mention that this package also comes with a web type interface for adaptive population selection designs. This web application features an easy-to-use graphical user interface, and you can launch it on your computer if you prefer a graphical user interface to the standard programming interface where you need to write and submit our code. And the last bullet point on this slide includes a link to the technical manual with additional information about the adaptive design framework that we have assumed when we were designing this particular package or this particular web application. This function for designing trials with data-driven population selection will be introduced using the following case study. It is labeled C1 in the package's documentation, and it is based on a confirmatory Phase 3 trial with a time to event endpoint. We will now take a closer look at this case study. I provided a detailed description of this case study based on a trial for the treatment of advanced non-small cell lung cancer back in part 9. It is based on the Saturn trial, and these two slides only present the most important features of this case study. This is a two-arm clinical trial where the single-dose regimen of the experimental treatment will be compared to placebo or standard of care. The primary endpoint in this trial, as I said, is a time to event endpoint. It is defined in terms of progression-free survival, which means that we assess the efficacy profile of the experimental treatment by measuring how it prolongs the time from patient's randomization into the trial to disease progression or death. Probably the most important feature of this trial is the fact that it relies on a biomarker-driven design, which means that the trial utilizes a multi-population tailoring approach. We talked about it in part 9. The patient population in this trial is assumed to be quite heterogeneous, in the sense that the efficacy profile of this experimental treatment is expected to be different in a subset of so-called biomarker-positive patients versus biomarker-negative patients. A strong beneficial effect is expected in patients who are classified as biomarker-positive, and the reason for that is that the experimental therapy's mechanism of action is closely related to this particular biomarker. The biomarker, as you can see on the slide, is based on the epidermal growth factor receptor, or EGFR, and the investigational therapy is an EGFR inhibitor. So this heterogeneity needs to be accounted for in the trial design, and this is the reason why we're considering this three-stage adaptive design with two interim assessments. The details of the decision rules that will be applied and the individual decision points are shown at the bottom of the slide. And the first interim analysis focuses on the futility assessment in the overall population of patients in this trial. And I would like to add that, at least theoretically, futility stopping rule could also be formulated and applied for the subset of biomarker-positive patients. But in general, a fairly small number of primary events or patients with disease progression is expected within the subset to be able to support a reliable futility assessment. And the standard method based on conditional power will be applied, and the trial will be terminated for futility if conditional power or predicted probability of success in the overall population of patients at the final analysis is too low. And if conditional power is sufficiently high, then the trial will continue to the second interim analysis. And this interim analysis supports an option to select the best population or populations for the final analysis, and the population selection rule will be presented on the next slide. The rule for population selection or hypothesis selection for the final analysis will be set up using the influence and interaction conditions. I introduced those two conditions and the associated decision-making framework in Part 9. And to give you a very, very brief description, the two conditions are designed to support a logically consistent way of defining selection rules and statistical inferences for general multi-population settings. In the context of this particular case study, the influence and interaction conditions will be applied to help the trial sponsor pursue the following effectiveness claims at the final analysis. As we can see on the slide, the sponsor may select the hypothesis that will help make the following claims. The claim of a beneficial effect in the overall population of patients, which is known sometimes as a broad claim, a restricted claim of effectiveness within only the subset of patients with a biomarker-positive status, and the last bullet point describes an effectiveness claim in both populations simultaneously. And here I would like to add that here we're using the terminology commonly used in pharmaceutical drug development, and in the context of more general randomized studies, we could simply replace the word claim with the word conclusion or scientific inference. Another point I would like to make here is that the last claim, the effectiveness claim in both populations simultaneously, is known as the enhanced claim. And to justify this claim, the sponsor needs to provide evidence of a beneficial effect across the overall population of patients. And also, that's the second condition, also to demonstrate a strong or enhanced beneficial effect within the biomarker-positive subpopulation. This would imply that the investigational treatment works for all patients or all comers, as they're commonly known, but biomarker-positive patients are much more likely to respond or to benefit from this new treatment. We now have all of the required information in place to begin the next subsection, and I will walk you through now the process of defining parameters required by the ADPOP cell function from the Mediana Designer package, as well as the process of running clinical trial simulations to be able to quantify the performance of the proposed adaptive design and to compute its main operating characteristics. After we load the Mediana Designer package, we're going to set up an empty list of function parameters, and over the next several slides, we're going to populate those required parameters. The first required parameter, just like in other examples we went over back in parts 6 and 8, is the endpoint type. As I said a few minutes ago, the primary endpoint in this case study is measured as the time from patient's randomization to an important event, which is either disease progression or death, and therefore this endpoint type would be time to event. The experimental therapy is expected to prolong survival as well as time to disease progression, and therefore the direction of favorable effect here would be higher. Larger values of PFS times would correspond to a beneficial effect. The last parameter on the slide specifies the number of enrolled patients in the control as well as the treatment arms. In this case, assuming a balanced design, 480 patients are expected to be enrolled in each trial arm in this case study. In the context of multi-population trials, it is very important to quantify, to specify the relative prevalence of biomarker-positive patients compared to the overall population, and this is captured by the prevalence parameter. In this case, based on historical data, the prevalence parameter will be set to 0.5, which means that half of the patients in this patient population are expected to have a biomarker positive status based on the predefined biomarker, which was based on the EGFR assessment. The second parameter on the slide specifies the target event count at the final analysis. This is a parameter which corresponds to the sample size or the target number of patients, evaluable patients, in a trial with a binary or continuous normally distributed endpoint. In this case, given the nature of the primary efficacy analysis, we need to specify the number of events that will need to be accrued by the final analysis. And given again the multi-population nature of this trial, we'll need to specify two different numbers. The first one, which is in this case 700, would be the target number of PFS events in the overall population. And if a decision is made at the time of the second interim analysis, is to only pursue an efficacy assessment in the subset of biomarker positive patients. In that case, of course, we will need to have a target for that particular subset. In this case, the study will continue until 300 events have been accrued within the subset of biomarker positive patients. The next two parameters define the efficacy expectations, efficacy profiles for the primary efficacy endpoint in the control arm as well as the treatment arm. In this case, it is very common to specify the median time, the median PFS time in this case, and given the anticipated heterogeneity of the treatment effect across the patient population, we need to have a separate set of specifications for the biomarker negative subset and for the biomarker positive subset. And that is the reason why when you look at the first parameter here, which is the control time, that is a vector of median PFS times in the control arm. The first number corresponds to the biomarker negative subpopulation, and the second one corresponds to the biomarker positive subpopulation. In both cases, based on historical data, based on the information about this particular biomarker, we expect the median PFS times, both values, to be 11 months. Now, continuing to a similar set of specifications for the treatment arm, we can see here that within the subset of biomarker negative patients, the median PFS time is expected to be prolonged from 11 months up to 13 months in the biomarker negative subset once again. And if we now continue and look at the complementary subset of biomarker positive patients, there we expect to see a stronger beneficial effect. This would be a good example of what's known as a differential treatment effect. We expect to see a certain amount of efficacy for patients with a biomarker positive status, but much stronger efficacy profile in the subset of biomarker positive patients. In this case, The median PFS time for biomarker positive patients is expected to be 15.7 months. And as it is frequently done, whenever we review assumptions for median survival times, it is helpful to quickly compute the implied hazard ratios. It is common to run those quick calculations assuming that the time to event follows an exponential distribution, and under this fairly reasonable assumption, the hazard ratio in the subset of biomarker negative patients is 11 over 13. It would be almost 0.85, which I guess would represent a borderline clinically meaningful result. But by contrast, when we now look within the subset of biomarker positive patients, where the treatment is expected to be more effective, the hazard ratio is equal to 11 over 15.7, or about 0.7, which does suggest a fairly strong beneficial effect. Specifications for the information fractions at the first interim analysis and the second interim analysis, as well as the parameter, the threshold for the futility assessment at the first interim analysis, are given on this slide. The vector of information fractions shown at the top of the slide has three elements. The first one, of 0.2 or 20%, gives us the number of events expected at the first interim analysis within the overall population. That would be the main goal for the first interim analysis. So within the overall population of patients, we expect to accrue 20% of the total target number of events, which happens to be 700 if we, as we specified it a few slides back. At the time of the second interim analysis, the information fraction is set to 0.6, so it's a slightly later interim assessment, and this is where we expect the milestone for this interim assessment would be accruing 60% of the total number of events in the overall population, and as always, the information fraction at the final assessment is set to 1. All the information will be available to us at that time. For the futility threshold, it will be set to 0.2 or 20%, which means that if conditional power within the overall population of patients is below the 20% threshold, then a decision will be made to terminate the trial for futility. This slide defines the thresholds used in the influence and interaction conditions as part of hypothesis selection at the second interim analysis. And very quickly, I would like to remind you how those two conditions are set up. Beginning with the influence condition, this condition is used for justifying the effectiveness claim in the overall population of patients, and this condition states that this broad claim of effectiveness in all patients across the entire patient population can be pursued only if an overall beneficial effect is not limited to the subset of biomarker positive patients. If this condition is met, then the next step will be to evaluate the interaction condition. And this condition deals with the differential treatment effect. I briefly alluded to it a few slides back. And according to this condition, the treatment is expected to be simultaneously effective in both populations, and therefore the enhanced claim could be made if the treatment effect in the biomarker positive subset is meaningfully greater than the biomarker negative effect. In this case, I'd like to remind you that we're dealing with a time to event endpoint, and the effects would be defined in terms of the negative log hazard ratio. That's a standard way of defining effect sizes for time to event endpoints, such as the time to disease progression. And in this case, the influence and interaction thresholds are set as shown on the slide. The influence threshold is 0.1 and the interaction threshold is 1.3. The next set of parameters describe the patient enrollment and dropout processes. The enrollment period here is expected to be 18 months long, and the median enrollment time, which is the milestone by which 50% of the patients will be enrolled in the study, is expected to be 13.5 months, which, as I've explained before, means that the first half of the patients will be enrolled It will take about 13.5 months to enroll the first half of the patients, and the remaining 50% of the patients will be enrolled between the 13.5-month milestone and the end of the patient enrollment period. As far as the dropout rate is concerned, in this case, it is very common to assume that the time to patient dropout, the time to the last contact for a patient who is lost to follow-up would follow an exponential distribution. That's why I'm here referring to an exponential dropout distribution. And what we see here is the annual dropout rate. So 5% of the patients are expected to be lost to follow-up on an annual basis. Now continuing to the last slide that describes the parameters required by the AD-POP cell function. We're going to specify the one-sided type 1 error rate, or alpha level, which will be set to the standard 0.025 level, and we will use 10,000 simulation runs to assess the operating characteristics of the proposed adaptive design. We're going to run simulations by passing this list of parameters to the AD-POP cell function. It's going to generate an object that is going to include all of the relevant information, all of the parameters, all of the assumptions that we've made, as well as the simulation results. They will be saved to this results object, and then we're going to generate a detailed simulation report by invoking the generate report function. The report will be saved in a Microsoft Word format, and a file called casestudyc1.docx will be created. If you would like to, you can easily download this R code as well as the simulation report created by this function, and you will find the R code and simulation report if you follow this link. We're now ready to talk about the key operating characteristics of this complex adaptive design with the futility assessment at the first interim analysis and population selection at the second interim. The most important operating characteristics are included in the simulation report. This slide shows the title page of the report generated for this case study. And just like before, the report is a Word document with a detailed summary of all of the parameters we've just specified. These parameters are listed in the first 8 tables, and you'll also find a summary of the simulation results in the last 3 tables, tables 9, 10, and 11. And the main simulation results and findings for the proposed adaptive design will be summarized on the next 2 slides. We will begin with a summary of key characteristics for the decision rules that are applied at the 2 interim analysis. This is table 9 from the simulation report. It shows the probability of terminating the trial for futility or lack of efficacy at the first interim analysis. I would like to remind you that this futility stopping rule is set up using conditional power for the overall population of patients. And we see here in the very first row of this table that this probability is actually quite high. It's over 15%. And that's where I would like to go back to the arguments I presented at the end of part 6 when we talked about operating characteristics of adaptive designs with sample size re-estimation. At that time, I pointed out that if we deal with investigational treatment, which is assumed to be effective, we would normally expect to see a lower likelihood of futility stopping or stopping the trial due to lack of efficacy. And therefore, even before we review the probability of success results that will be presented on the next slide, we could predict that the trial may be underpowered. In cases like this one, I would recommend considering lower values, lower thresholds for conditional power at the first interim. We set it to 20% a few minutes ago, and it would make sense to lower this threshold and run additional simulations. Again, this would be an example of an iterative process of designing trials with complex decision rules. So this was a comment related to the first row in this table, and the other 3 rows in table 9 from the simulation report give us the probabilities of population selection for the final analysis. When we look at the efficacy data, PFS data at the second interim, the predefined rules based on the influence and interaction conditions will be applied. to identify the most meaningful hypotheses, one or potentially two hypotheses that will be tested at the final analysis. And this table shows us that most of the time, almost 44% of the time, both hypotheses of no effect in the overall population and the biomarker positive subset will be selected. Here I'm referring to the bottom row of this table, and this is the most likely outcome for the second interim analysis. Less often, single hypotheses in the overall population, the biomarker positive subset, will be chosen for the final assessment. Those probabilities are given by 26.4% for the overall population and the 30% for the probability of selecting only the subset of biomarker positive patients. This slide combines the results including tables 10 and 11 in the simulation report. These results support an informative comparison of the traditional single population design, which serves as a benchmark, and the multi-population design with adaptive population selection. I would like to mention here very quickly that in the traditional setting, a futility stopping rule in the overall population would also be enabled. This will be done to support a fair comparison of the two trial designs, the two trial approaches, but the final analysis in the traditional study will be performed only in the overall population because once again it's a single population design, and it follows from this table that both approaches for trial design, traditional and adaptive, would guarantee about the same level of power, about 80%. Here I'm comparing the top row with the bottom row, and the values I'm referring to are shown in boldface. And in addition to that, for the adaptive approach, the table also gives us the probability of success within each individual population, overall population or the biomarker positive subpopulation. This information is generally helpful, but of course it would be of secondary importance. And the main conclusion here is that adaptive multi-population designs with population selection provide an appealing, flexible alternative to traditional single population designs. This was the last video. We're done with this training program. I really appreciate this opportunity to tell you about our work on simulation tools for adaptive designs. Thank you so much for your interest in this general topic of adaptive designs. I would also appreciate feedback. If you happen to have any questions, any suggestions, any feedback, please feel free to contact me. You see my email address on this slide. Thank you very much.